guys, thanks for joining me, and we're going to learn about the cell today and look at chapter 3. So as we learn the various organ systems, their functions, and how they interact with each other in both A&P1 and 2, we'll need to discuss what is going on at the cellular level. The cell is the basis of life, and with that in mind, we need to study the processes that are going on at the cellular level that allow the organ systems to carry out their functions. It doesn't matter what we're studying in A&P, whether we're studying digestion or the muscular system, respiratory or nervous system, or even the urinary system. All of these processes are cellular processes. So with that in mind, let's take a look at the cell. Now, I have already given you a peek into a day in the life of a cell in my chapter 2 video, but here we will go further. So here are the most important topics that you will need to understand from chapter 3. Some of these topics I will explain further, and some you will need to learn on your own. There are other required videos that explain some of these topics really well, such as the video on membrane transport that comes from your textbook. That video covers membrane transport in the exact detail that I would like for you to understand, so make sure that you pay attention to it and take notes. The majority of the processes that we will study from chapter 3 are happening at the membrane level. So we will begin by looking at the structure of the membrane and the fluid mosaic model. What is important to understand about the membrane is that it is a dynamic structure whose components are in a constant state of motion in a sea of fluid. And as discussed in our chapter 2 video, the two main components are the phospholipids and the proteins. Let us now look more closely at the phospholipid without getting too far into the chemical detail. Here we can see the phospholipids arranged with their hydrophilic heads aligned toward the outside and inside of the cell, and that is because the heads are polar or charged and are attracted to water, whereas the hydrophobic tails are aligned with each other within the middle of the membrane because they are repelled by the water. This causes the outside and inside surface of the membrane to attract charged particles while the middle section repels them. Now let us look at an animation that explains how this configuration affects the movement of substances across the membrane.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
Thank you, Molly, for that very informative animation. Now, another very important component of the membrane are the various proteins that are studied throughout it. It is the proteins of the membrane that really control the activities of the cell. These proteins have numerous functions, and it is a good idea to understand the various functions that they carry out. Here is a good summary taken from your textbook. Make sure that you know these functions. Just a few words now about the glycocalyx. The glycocalyx is a sugar coating on the exterior surface of the cell membrane and is composed of highly specific biological markers that allow cells to recognize each other. The ability of our cells to recognize each other prevents our own cells from being attacked by our immune system. A failure in this ability can lead to such things as autoimmune disease and infertility, for example. You will be needing a basic understanding of fluid dynamics, which is the movement of fluid and air through the body. This is a very important topic in relationship to the clinical concepts that you will be applying further on down the road. The movement of fluid through the various compartments of the body is a closely monitored system that involves numerous organ systems. We will be studying these different compartments in the fourth week of school. But for right now, we are considering the extracellular, also known as the interstitial fluid compartment, and the intracellular fluid compartment. Please spend some time with the following topics in your textbook and watch the posted videos on membrane transport and the magic of osmosis before returning to this video. And I'll be here when you get back.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypotonic environments, water rushes into the cell, and the cell swells, but is kept from breaking by the rigid wall layer. The pressure of the cell pushing against the wall makes the cell turgid and is the desired state for most plant tissues. For instance, placing a wilted celery stalk or lettuce leaf in a hypotonic environment of pure water will often revive the leaf by inducing turgor in the plant cells. Animal cells lack rigid cell walls. When they are exposed to hypotonic environments, water rushes into the cell and the cell swells. Eventually, if water is not removed from the cell, the pressure will exceed the tensile strength of the cell and it will burst open, or lyse. Many single-celled protists living in freshwater environments have contractile vacuoles that pump water back out of the cell in order to maintain osmotic equilibrium and avoid lysis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypertonic environments, water rushes out of the cell, and the cell shrinks away from the rigid wall. These cells are dehydrated and lose most or all physiological functions while in the shriveled state. If cells are returned to isotonic or hypotonic environments, water re-enters the cell, and normal functioning may be restored. Animal cells lack rigid cell walls. When they are exposed to hypertonic environments, water rushes out of the cell, and the cell shrinks. The resulting cells are dehydrated and lose most or all physiological functions while in the shriveled state. If the cells are returned to isotonic or hypotonic environments, water re-enters the cell and normal functioning may be restored.
Thank you, Molly, for helping us understand osmosis and tonicity better. We will be discussing all of these topics further in class, so make sure you have a good grip on them and bring your questions. The topic of resting membrane potential is historically a confusing one for students. The selectivity of the cell membrane, along with the active pumping of charged particles into and out of the cell, produce a difference in charges or a potential across the cell membrane. All cells have a resting membrane potential. However, it is the nerve cells and the muscle cells that depend on this potential for their function. We will explore this topic in greater detail in their respective chapters. For now, it is only important that you know that cells have a resting membrane potential. Our final topic for this video are the cell organelles. Please know the organelles of a cell, know how to recognize them on our models and pictures, and know their basic functions. I have summarized the functions of the organelles that you need to know and will present them to you now. The mitochondria, the power plant of the cell providing most of its ATP supply. Ribosomes, sites of protein synthesis. Rough endoplasmic reticulum, membrane production and protein packaging. Smooth endoplasmic reticulum, synthesis of lipids, detoxification of toxins, and breakdown of glycogen. Golgi apparatus, traffic director for proteins destined to exit the cell. Peroxisomes contain enzymes that neutralize free radicals. Lysosomes contain digestive enzymes and are the demolition crew of the cell. The cytoskeleton, which contains filaments and tubules, which are the bones, muscles, and ligaments for the cell. The centrioles form the spindle during mitosis and form the base of cilia and flagella. We have the nucleus, which contains the genetic library, and the nucleolus that produces the ribosomal subunits. Within the nucleus is the chromatin, which is the cell's genetic material. And lastly, we have the cellular extensions, the cilia, flagella, and the microvilli. The cilia move substances across the cell's surface. The flagella allows cells to propel themselves. And lastly, microvilli are finger-like extensions of the plasma membrane, which increase surface area. That ends the material for Chapter 3. Make sure that you try the few end of chapter questions that I've highlighted here.